the recording. Here we go. And the floor is yours. Thank you very much.
Thank you very much, Camille and Nicolas, for your introduction, and most of all for your support the last 5 years, and also now for connecting our project. So we will start to share the context that motivated our project, and then we will provide some details about the data we have collected and make available to the community. Next, we will provide you some example of results we have done. And finally, we will introduce the infrastructure of FISHGLOB we want to deliver this forthcoming years. So first of all, our project is linked to the effect of global change of fish diversity, more specifically on demersal fish, so the species which are linked to the ground of ocean and sea. And so global change is linked to several sources of impact, and one impact that we are more focused on is the climate change, so change in temperature, and also the fishing pressure. So about the first one, the climate change, we know that nearly 70% of the ocean show increased human impact, and mainly driven by this kind of climate change pressure. And this shift in temperature can lead to a change in species distribution and composition. It is mainly known for surface marine heat wave, but there is also some bottom marine heat wave that can occur until 4,100 meters depth. And the effect of this kind of marine wave is not really known on fish community yet. So we are interested to assess this kind of impact. So here, for instance, you see different maps of regions in the US, in Northern America, and the various intensity of bottom marine wave, and with an average increase of temperature. You can see that it can reach until 5 degrees in addition to the normal temperature. And these bottom marine wave are sometimes longer and more intense than the surface marine wave. So surface marine wave are considered usually until 30 meters depth. And usually they co-occur, both bottom and surface marine wave, but sometimes there is only bottom marine wave. So we are very interested to know also the change of these bottom marine waves. So previously, the impact of marine heat wave, but also change of temperature more in general, have shown some impact on species range shift in fish at regional scales. And more globally, we predict that at the global scale, there will be important change in fish composition that will lead about 40% in tropical area change of diversity. So here you have two maps, and the first map at the top is about the rate of change of species invasion, so new species, and the bottom species extinction. And on the left, you can see the percentage of species turnover according to the latitude. And so the percentage, the predicted percentage, with the most pessimist climate scenario reaching about 40% of change. So we are very interested also to address this kind of research question, the impact of temperature on the diversity and species composition change. And this range shift of species can have an important impact also on stock, on transboundary species, more particularly, because it can change the catch proportion of commercially species that can be caught by industrial fishery or also artisanal fishery. And for the coming years, it is predicted that in 2030, about 33% of stocks will have moved, and that almost 80% of the economic exclusive zone will have at least one stock move. And this kind of change of distribution can lead to conflict among countries for resources, as it had already been observed in the past for several regions. So there is also a very important issue in terms of fishery, industrial fishery to address with this. And also, all these topics are very linked to ecosystem functioning and services, and because fish are known to contribute to carbon sequestration and also to provide a source of proteins and micronutrients to human populations. But what we know also is that the effect of global change is not homogeneous in space and time across the globe. You can see on the left a map of the sea surface temperature, annual mean sea surface temperature, and on the right, the distribution of fishing effort. And as you can see, it is not homogeneous across the globe, meaning that if we want to have a better understanding and to better predict the change of fish diversity and composition across space and time, we need to assess the past, current, and future distribution in different areas across the globe. So to doing so, to assessing the global change impact on fish across the different sea and oceans across the globe, there is a different kind of data that have been collected in the past. And we focus on the scientific bottom troll surveys because they usually provide a long time series. We collected series that are longer than 10 years of duration. And these surveys collected individual counts of fish, but also the biomass and in some cases size also. And of course, the main feature is that they do not use the same strategy. They don't have necessarily the same temporal and spatial extent. They don't mainly focus on the same species sometimes. But even with this feature, we can address some really interesting scientific question by studying the relative trend in time between these different regions, these different surveys, in order to deepen our understanding. And there is also various status of accessibility of this data. Some of data are public, some other are private. And so we try to make an overview and inventory of the accessibility and status of this data. So the main aims of FISHGLOB was first to create a network of expert and data provider on scientific bottom troll survey across the globe to make an inventory of the existing surveys and the related metadata, that is to say, in which year, which location, which kind of data collected, and then also to collect the full data, meaning the biological data, so the counts and biomass of this data, and to provide an homogenized format of data after a precise specific processing of data to make a unique format of data. And also to provide to the scientific community the contact for private data in order that people can directly contact data provider, in that case, for asking some collaboration, because we cannot share directly this private data. And of course, finally, based on this data, we wish to address some key research questions related to the global change impact on fish population and community. And so finally, the main aim is to provide an integrated database and also an infrastructure to make the link between science, conservation, and also fishery management. So just to talk about how FISHGLOB was created, at the very beginning, in 2019, it was an idea that had been shared between 5 scientists, among young scientists, so Aurore Moreau, Romain Frela, who is now a data scientist at CESAB, Lorraine Pécuchet, and also James Thornton and Nancy Schachtel, who did a side meeting in an ICES conference. And they made the idea to provide an inventory of bottom troll survey and collect metadata. And then from this idea, we have created a bigger group in order to collect the full data and to start to address research questions. And to do so, we answer to a joint synergy call between the CESAB and the CEIA, which is the equivalent in Canada of the CESAB. And this call allowed to found two in-person meetings, one in France and one in Canada. And more recently, so we have started under the umbrella of the United Nations Decade of Ocean Science to build an infrastructure for FISHGLOB in order to make the link between scientists, data provider, and fishery management. So this is still ongoing, and we will see in the presentation how we have built this. Also to share that the initial and core FISHGLOB statistics group was mainly being part of a researcher from Northern Canada, Northern America, Norway, France, and that from this group we have now growing and created a network of data provider and experts, which reach more than 100 people now. And we have a steering committee of more or less 11 people to organize all the projects. And we have also created different data sets, metadata covering at least more or less 100 surveys, and also full data set of almost 30 surveys. So now we are going to share different activity we have made in the frame of FISHGLOB. First, then we will present the data sets and how we create our community of experts around the fish community, and the current status of the infrastructure. Then we will present you two main scientific studies in terms of research we made linked with the temporal variation of fish community and with a focus on the effect of marine heat wave. And finally, we will present you the perspective about the infrastructure of FISHGLOB we are starting to build and where we want to go. So then if you want to take the floor.
Yes, thank you. Thank you, Bastien. And thank you, Nicolas and Camille and everybody at Cesab who had supported the FISHGLOB community since it started in 2019. That's a long time ago. And then we have 2020 when we did everything online because it was COVID. Yeah. So I will talk to you about the FISHGLOB datasets. So this is what FISHGLOB datasets are. So if we can see from this schematic presentation, we can have the scientific survey types from different types of gears and different types of surveying methods, right? So this gives you an indication of how wide the survey data types are. And from this, Aurore and Giuliano, another member of our team, and Zoe, were able to synthesize the data sets into unique surveys, like with survey metadata by institution, gear type, and contact information. That's what we have as a survey data. And then there, further down that line, you have the event metadata and standardizing the event metadata by using all this structure, right? Unique ID, date, time, effort, and location. And then further down is the going into the data itself. That's with this identification, unique observation ID, taxon number, and weight. Not all of these data sets are open. And therefore, what Aurora and Giuliano did was to categorize the types of data sets that are there. So if you had an open survey and if the survey data is not completely open and only this metadata is open, that is your first category there. And then you have the open survey and event metadata. That means we can only we can only use the metadata and event metadata. And then there are data sets that are completely open, and that is the third category. Next, please. Now, when they were processing the data so that they could standardize it and homogenize everything in a format that could be analyzed, they had to look at the format survey, homogenize everything across all surveys, and then there is cleaning of the taxonomy because the names might not be the same throughout all of the surveys, and you can have common names being used in one survey and scientific names being used in another survey, and so on and so forth. And making sure that all of the scientific names are valid currently. Then there was going backward and forward with experts. And so there was feedback from experts that was necessary during the survey data processing because there was stuff that were not clear. And then, of course, the flagging of taxonomy issues in the time series. Next, please. The metadata inventory and a network of experts. So this subject was published by Aurore in 2021 and describing the whole data set. So there were 95 bottom trawl surveys that covered the period between 2001 and 2019 with 283,000 or almost 284,000 holes or samples, and of which 59% are not publicly available. Next slide, please. The full data of publicly accessible surveys covered 29 public surveys. That was integrated into the open FISHGLOB database that is available in GitHub. Then this covered 18 countries. As you could see in the map, this is everywhere where the data is fully accessible, and covers the period 1963 to 2021. And these are made up of almost 2,200 sampled fish taxa, and just a little bit over 216,000 hauls. Next, please. Let's say the people who are using or who provided and used and who are going to use this data. This is our let's say that mantra is that we respect all the data sovereignty and collaborations, right? So the open data and open science is our guiding principle. Data sovereignty also is respected because this is a very niche type of research. We need to build capacity not only for people who are going to use the data, but people who are going to add to the data sets so that we can grow an international user or let's say network of people using and providing data. And of course, we believe in credit and visibility for all consortium members. The consortium, when we say consortium, this is not only us, the people who developed FISHGLOB, but also all of the data contributors, because without them, there will not be FISHGLOB. So that's the thing. We would like to build visibility for those people who actually did the surveys. Next, please. And FISHGLOB abides by the FAIR principles. And if you took a picture of this QR code, you will be able to look to get into the GitHub and the website for FISHGLOB. Next slide, please. So now we are transitioning into the first of the two major studies that were published while once the data sets were ready for analysis, after the work of Aurore and Giuliano in standardizing and homogenizing all of the data sets, Zoe Kitchell, sorry, I have to read my notes. Zoe Kitchell leveraged the wide spatial and temporal coverage of FISHGLOB data, in addition to additional 12 survey data from collaborators, to ask the question of what are the impacts of species range shifts on regional community composition. Now, once the potential outcome of these asymmetries in how animals in the ocean are responding to change is, next slide, please. The biotic homogenization, where across space, communities start to look more like another in species composition. That is, they are losing their individual uniqueness. Homogenization occurs as fish communities begin to look more and more like one another. Next slide, please. In contrast, another potential outcome is biotic differentiation, where across space, communities that start to look less like another. And that is, these communities are becoming more unique. Now, both homogenization and differentiation can impact on ecosystem stability and resilience. Next slide, please. For this study, Zoe worked alongside collaborators and data providers from many countries to compile a data set with 34 unique surveys that covers the period 1968 to 2021, with over 2,000 finfish species and containing information on species biomass for over 178,000 tow events. Next slide, please. Zoe calculated the Jacquard dissimilarity indices for these data sets. That is, for if you look at the schematic diagram, homogenization might be indicated if the index is trending down across the time series, or differentiation is indicated if the index is trending up during the time series. So, for instance, here, the example is Southeast USA in the West Atlantic, which is apparently experiencing homogenization, whereas Ireland, oh no, not Ireland, New Zealand, west coast of the South Island, is experiencing, as you see, the index is going up and is experiencing differentiation, whereas in Iceland, there is no trend through time. Next slide, please. Across all of these 34 surveys, Zoe identified 10 that were homogenizing, 11 that were differentiating, and 13 without any significant directional change. There are no clear patterns by area, time series length, or number of species were found, but both differentiation and homogenization occurred in the northern hemisphere and only differentiation in the southern hemisphere. But Zoe had access to more surveys in the northern hemisphere. So might be an artifact of the fact that there are more surveys in the northern hemisphere. Next slide, please. Oh, that was in press. Is it still in press, Bastien? This work is still in press?
Yes, it has been accepted 1 week ago.
Ah, yeah, that's right. That's why I thought it was already published. Anyway, so there are no, as Zoe found out, no overall relationship with bottom temperature, but there are strong regional patterns. For instance, on the Rocktail Plateau, where you have it on the inset, the warmer years were more differentiated. So the communities during the warmer years were more differentiated. So that's where the arrow is pointing to. Next slide, please. Now, in the north, so we also found that there were no overall relationship with fishing pressure, but again, there are strong regional patterns. And for instance, here that are with the black arrows in the northeast US, years of higher fishing pressure were more differentiated than the rest. And so it's a very interesting thing because it means that there are regional patterns that we have to look into further. Next slide, please. So her conclusions are that on species composition changes, there are dynamic patterns of homogenization and differentiation over time, and that homogenization and differentiation are no more common than no trend. And then impacts of temperature and fishing pressure are context dependent, with temperature temperature may not have a direct or indirect impact on fishing. Then there is homogenization it's instability. It could be a signal for instability, and the more differentiated fish communities can help sustain regional fisheries.
So
And now this is Bastien. Yes.
thank you, Leng. So in addition to this work dealing with the change of fish community across time and space, an additional research that have been led by Alessa Freston from US is the effect of marine heat wave on fish community in the continental shelf, meeting until 200 meters depth. And so we focus in that case of the public data also in the northern hemisphere by studying the effect of almost 250 marine heat wave effects. And so we assess the effects at the community scale from different variables. And so here we made just a focus on the biomass. And so on this slide, you can see the biomass log ratio between the biomass after and before marine heat wave. And so what we can see is that in some cases, we see that marine heat wave led to lower level of biomass. And it can be until less than 80%. But in general, from all the survey we investigate in the northern hemisphere, by taking into consideration the 250 marine wave, we do not see a general effect of marine heat wave on fish biomass. There is also no general increase in the variation for the absolute biomass log ratio according to the intensity of marine heat wave. And in fact, in addition to the effect on biomass, we also analyze different response variable. For instance, an index that take into account the thermal affinity of species. We also assess the community variation with dissimilarity index, the abundance data also, some traits, the effect of different depth, effect of latitude, etc., etc. And in any cases, we don't find a general effect of marine heat wave on demersal fish diversity. So the main explanation could be that the fish can move maybe to a deeper habitats, and that can provide some climate refugia. And so that can be led to a short-term resilience. But finally, we don't know this resilience, if it can be different according to the intensity in terms of duration of marine heat wave. If a marine heat wave lasts several weeks, maybe the resilience could not be the same of fish. And this because we know that although the marine wave becoming more regular, more intense, more longer, finally, we don't know yet the limits of marine heat wave resilience for fish, even if in this case we don't see any effect. So the main conclusion of what we have presented until now is that the FISHGLOB allow to create a new international scientific consortium of experts and data providers, and to build from a regional survey a global data set after a specific process to provide a unique format to the community. And in terms of science, we have seen that community did not show any differentiation or homogenization or marine heat wave impact as a main rule on demersal fish community, at least for the time window and the survey we have investigated. We also provided a new method to input species trait data when they are missing in some species group. So it is a method that is also useful for any terrestrial species, not only for marine species. So we didn't present this work, but we think it's quite interesting because of the very broad applicability of this method. And also to share that the FISHGLOB data have been already used by the scientific community to address some research questions, notably the three work we mentioned here. And so the challenges and feedbacks we can provide to you in coordinating FISHGLOB is that first we start FISHGLOB exactly when the COVID started. So it was quite difficult to manage because initially the funding we have from CESAB and CII was for 2 in-person meetings. So in fact, we translate it as regular video meeting. And also with the help of CESAB and CII, we could contract Giuliano Palacios Abrantes to help us in the data processing, which was a very huge task. And also, what we found specifically for 1 year ago is to find the sources of fundings to support an infrastructure and online platform, because there is very few sources of fundings. And so if you are in such cases, if you want to start a CESAB project or data share CESAB project, I think it has to be very anticipated because we have already tried 2 different sources of fundings and we have a negative answer. And we have tried to US fundings, Monaco fundings, but there is not so many fundings available. Now we are waiting for an answer for European projects, so we will see if it's accepted or not. And we try again also in the US. But given the context, we are not sure to get the fundings because the thematic is really linked to climate change. So we will see. And so also to share that now, and also since 2023, we are under the umbrella of the United Nations Decade of Ocean Science. And the aim is to create a sustainable infrastructure together the different experts around the demersal fish community, and to provide link with governmental agencies and stakeholders because we are directly in link with fish community of commercial interest. So it has also a very applied aspect. And so the idea is to provide some data and also some baseline to understand and predict the diversity change of this community to guide management action. And so now FISHGLOB is constituted of a network of experts, and so more than 100 people, as we said at the beginning, a steering committee of 11 people, and also we are structured in four main working groups. First, around the activities to the consortium building, the infrastructure, the research aspect to coordinate this, and also different application towards the management and stakeholders. And so now, then we'll present you more in detail the infrastructure we are building, which we are trying to building. We start building some things, but there is a lot to do in the forthcoming years. So then, if you want to take the floor, please.
Yeah. So this is a schematic presentation of our diagram of what we want, what we're trying to do. So we have FISHGLOB at the center of a network, and we have data contributors, we have associations with large databases that are out there, and trying to also use certain applications, for instance, Life and Ocean Adapt, Fish Base and Sea Life Base. Next slide, please. Next slide, please. So let's look at the FISHGLOB association or relationship with Fish Base and Sea Life Base and Fish Life. So Fish Base and Sea Life Base are the sources of species-specific life history traits, but some of these traits are not available for all of the species that are sampled in the troll surveys, and therefore Fish Life is being used to impute traits of species or groups of species that this is actually described in another publication by Thorson et al. In 2023 with FISHGLOB data that do not have traits data in Fish Base and Sea Life Base. Next slide, please. Now we are looking at the wider user community, which of the existing projects are going to be using FISHGLOB data. So via the European Ocean Science Cloud Services, we are also able to provide FISHGLOB, and we have data exchanges with other projects and other data aggregators, for instance, FishMIP and BioShifts. Now, within the research community, we situate FISHGLOB data for use in other research ideas and other synergies, for instance, with EOSC and other data aggregators, and among individual scientists and researchers who have the interest in using FISHGLOB data in their research. Next slide, please. Now, we are able to provide FISHGLOB in interoperable terms, right, with other data aggregators. And that means that FISHGLOB has to evolve as well, because if we're going to provide it with other data aggregators, for it to be interoperable, it has to be in the right format. And I think we had done a good job at homogenizing and standardizing such that FISHGLOB data is ready for instance, contributing to OBIS. And as I was saying, we have FISHGLOB is not just a simple species occurrence, because it has other information that, for example, fish traits or life history traits that are in that information system. But the goal really is to link open data that is built around the shared semantics and global identifiers. And we have prepared FISHGLOB to do that. Next slide, please. So this is the vision of one of our main, let's say, architects of infrastructure, infrastructure, cyber infrastructure. And this is coming from Rob Guralnik from, I forget where he's from. He's from the museum in Florida. And he devised this infrastructure such that we have the database that gives you all the data stuff, the data infrastructure. And then you have some value-added products that would be testing, for example, interpolation testing, and that would be a gap-free product development and the development of an R package so that everything can be put together and so on and so forth. But then the most important thing that Rob wanted to highlight is the community, the social infrastructure, the community of data contributors and the community of data practitioners, basically. So the end game is to build a community of practice who will not only use the data sets that we have, but also add into that existing infrastructure, into the data that is already there. So this sort of infrastructure not only makes FISHGLOB open to new data coming in, new surveys to be homogenized and standardized, but also having that community of practice established such that it grows and becomes really useful for everybody. There you go. Next slide, please. So if you have any interest in FISHGLOB, please just fill the form on our website. The links are here, and tell us what we can do better or how much we're doing well. And then, yeah. So thank you for supporting us, CESAB first, the CIE here in Canada, especially the French embassy in Canada, who's provided a big help for mobility of both Canadian and French researchers. Our other partners, University of Montpellier, Rutgers, HIFMB, and Thunen, and the sea around us, which is where I am from. Thank you.
Many
Oh,
thanks.
thank you. Thank you very much. Thank you very much for both of you for this amazing, amazing work. Now we have some times for questions. So people in the audience who would like to ask questions, please use the tool here to raise your hand, and I will distribute the turns. And if you're unable to speak, you can also just write your question in the chat, and we'll keep an eye out for them as well. So, does anyone have a question? Well, Nico, you're not using the tool, but I can see you, so maybe go ahead.
I mean, I will break the ice. Thank you very much. I mean, that was very, very impressive. The amount of, I mean, you really get that it's a human adventure before all. And I really like the way you try to build the ecosystem also, and to get in relation with what is being doing at a larger scale. So that's really good. I've got a scientific question, but I've got also I'm very curious about how did you end up being selected as a UN Decade Action, and did you apply for it? And also, did it actually open doors for you to get support? So just to get your feedback on that.
Yeah. So we apply. It is a process where you have several calls per year. I think you have 2 or 3 several calls. And so first we contacted the program, because we are a project and you can be under the umbrella of a program. And so it is the Supreme program led by the NOAA in the US. And so they're mainly interested in prediction of marine ecosystems. So we contacted them in order to know if they will be interested in our consortium, in our data to make common research. So they were interesting. So based on that, we applied to the UN Decade to be a program under the umbrella, a project under the umbrella of this program. And then once you have the label, it can open some doors, but For time being, it have been maybe useful for us to find some additional fundings, notably from the University of Montpellier, also to make an additional in-person meetings. But for the time being, I will say it is the only plus we had with the UN decade, because by themselves they don't have any specific findings. So it can help to get some, but from other sources. But from the time being, because we try to find some fundings to build the infrastructure, which is very specific, and there is not so many call, it's mainly through foundation we can get that. We still not have any additional support, I will say, from the UN label.
Okay. Blue cask, maybe.
Yes.
Well, anyway, sorry for that bad joke. Who else?
Well, nobody is raising their hands yet, Nico. So if you have a scientific questions, maybe.
Lisa.
Oh,
Lisa.
Lisa.
Yeah.
Thank you, Lisa. Go ahead.
Thanks for the presentation. So I arrived a little bit late, so maybe it has been covered, but I will still ask the question. You talked about shifts in community composition, but you also mentioned you collect traits. So did you also look into that?
Yeah, the traits that if you remember that there was a paper by Thorsten. There are two papers by Thorsten et al. That was published earlier, and then the latter one was published later. So it's there are traits that were collected, and using FishLife, which is Jim Thorsten, which is Jim's software. He was able to collect a little bit more traits for species that did not have the traits in fish base and sea life base. So, and those traits are available. It's like it's available in the FISHGLOB database, but I'm not entirely sure if it is something that is downloaded with the survey data. I think it is something that's apart, that's separate. Yeah. So, but it can also be obtained from FISHGLOB people. Yes. So yeah, the response is yes.
So if you want more detail about that, the paper we mentioned from Jim Thorson in 2023 in the method ecology and evolution, where he proposed the tree imputation methods. Yeah, we also made in that work an analysis for all the fish worldwide, so 20,000 species, the traits, and to assess the effect of temperature of several variables on traits. So there is some information also in this work, but it is this work is beside the FISHGLOB data. It's from the FISHGLOB group, but not with the FISHGLOB data. This one.
Yeah, because it's purely biodiversity. So it did not how to say that? It did not use the FISHGLOB data set per se. Yes.
And then at the community level, we have started also to address some question about functional beta diversity. In addition to taxonomic beta diversity, we presented the result from Zoe. We have also started to assess some question about functional beta diversity with traits. So it's at the very beginning, because we didn't have time to implement all, but it's in the pipeline. So we will try to work on that next year and the next year also.
It's the future sort of thing. Yeah. We're still planning, even if we don't have money yet.
Thanks.
Thank you.
I might have a question as well. So thanks for the presentation. So it's been quite a lot of effort to get all these data together. How to keep the database updated, how to keep things going, because once it's published, it's nice, you have a nice output, but then afterward it quickly gets outdated. So how you think of handling that?
Yes. Talking to somebody who's been 30 years in data database development. Yeah, Fishbase is 35 years old actually. So yes, so this is a question that our community of practice has been asking ourselves. And I mentioned that in the presentation for the infrastructure. And one of the things that we are planning to do is to have a way of updating the data. We don't know how that will happen yet because the surveys might be, as you remember, there were three categories of survey data. One that is only where you have access to the metadata, and then there's the others that are really completely open. And so to introduce that kind of complexity in data updating is a little bit difficult, and that's where Rob comes in with all of the digital infrastructure that we didn't show it, but that Rob has this idea of a digital infrastructure that would help let's say make it easier for the updating of the data sets. So, but we need money for that because we need, in that case, we need a data person, we need a database software engineer. So now we are going into the technical part of it, right? So yeah.
It is exactly the content of the project we have submitted to a Eurocall, I think two months ago, more or less, yeah. And so yes, it's really to create a platform and also a routine and pipe in order that we can update the current data we have. So already survey we have identified and work with it, but also to welcome a new survey we didn't have the opportunity to consider yet. So we have to try this year.
Yeah, because remember that there is the question of standardizing and homogenizing, so that it is in the same format, is in the FISHGLOB format for easier use. Yeah. Yeah, you need warm bodies behind, but right now we don't have those warm.
Maybe it is a question now from me. I don't know, from the CESAB, do you think that the data scientists, such as you, Romain, could have a role in this data maintenance or not?
I'm the one asking the question. Yeah, I don't know. We can discuss that. But yeah, that was a side remark that you also found a spot I heard in Oldenburg, right? Where you might meet as well again. How did that come up?
Yeah, in fact, during last December, we make an in-person workshop in Montpellier, the CESAB. And during that workshop, we made an half-day conference, and a guy from Germany, so Karl Weiner, asked if he can come in France during the workshop and the conference. So we welcome him at the CESAB. And a few weeks after, he have been in link with Helmut Inbrandt from the Functional Diversity Center.
HIMFB.
In Edinburgh, and who had the possibility for fundings to welcome us in Germany for a 1 week in-person meeting. So it means that in December this year, we will go for a meeting in Germany for our group. Yeah. And we will also welcome people from all our consortium, from 100 people of the FISHGLOB consortium to come if they are available and if they have their own fundings, because we are covered for the main group, the core group, the steering committee, but not for all the FISHGLOB people. Yeah. So yeah, it was really
thanks. Yeah, it's a good news to know that it's continuous after CESAB. Cool.
Yeah.
Nice.
And also it's continue Also with CESAB, because there is some new CESAB projects which are going to use the FISHGLOB data also.
Mm.
There is the FARSEX project. There is the new project from Olivier Maury. I don't remember the name, but
FISHMIP.
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 FISHMIP, the name also for CESAB is FISHMIP, okay. So FISHMIP, yeah. So yeah, we are linked also with BioShift project. So it's nice to see also the synergy there is among the CESAB group. Yeah.
Mm.
Indeed. Yeah, Nico, go ahead with your scientific question.
I'm totally naive on the heat wave, and I'm dreaming of a cold wave right now. And so, well, my question is about, how do you classify the heat waves? And is there, I suppose there are some kind of accepted classification, but that would be my first question. And my second would be that if that classification exists, why it is not based on organisms? Like, some fish, depending on the generation time, depending on the size, depending on many things, you're going to experience the heat wave completely different. And for some species, what we define as a heat wave might not be a heat wave. So I'm wondering if that mess doesn't explain the absence of tendency you got in your results.
Yeah, I understand. Yeah, so indeed there is some, I will say, standard definition of marine heat wave available in the literature based on the duration. So in that case, we use a 5-year duration of heat wave, and a heat wave is defined, is a relative definition based on the percentile of the temperature data. And so in that case, if I remember well, we took the 90 or 95%, meaning that we took the more extreme events, so the stronger marine heat waves. And so the second part of your question, indeed, I think an heat wave can be very species dependent. And so in that case, we assess the impact of marine heat wave also based on the species thermal affinity of individual. I didn't present this result I just mentioned with all the variable, but yeah, we try to assess this. Yeah.
Okay, and you looked only at the higher. You haven't looked at the cold wave.
No, we didn't make cold wave. Yeah, okay. Yeah. Yeah, but it will be also interesting to see if there is some change with cold. Yeah.
Yeah, I would say that if you have a delta of something, then you have to look at the high and the lows, and that's very interesting.
Yeah, but we are starting to do it in another work led by Laurent Pécuchet, who is making a study related to life history tree of species, and also the effect of temperature and different variable. And so we have also the lower temperature that is taken into account. So we will see in that case if we can see an impact of lower temperature. But it is not in that case based on the definition of a cold wave, for instance, where you could set it based on the duration and also the distribution of temperature. Yeah. It's the main variable of temperature, which is taken into account. So, yeah, maybe cold wave per se could be very interesting, yes, to see also.
Okay, well, I guess if that's all, we can call it an end of this Cesabinar. Thank you very much. It was all super interesting.
Thank you very much to you for
congratulations.
Thank you. Yes.
And thanks again to your support every time since the beginning, because now we have 6 years of existence. So thanks to you. So we're very glad for that, and to create our group, as you said, is a very beautiful human experience first. So we are very pleased to meet together, to work together, and to have built a more larger and stronger community around this scientific thematic. So thanks very much to you.
Thank you.